So the first group is the group of ceramic and natural nanomaterials. Pavel, are you here?
Yeah, I'm here.
Okay, great. So I will change slides, so just tell me next when you need to change it.
Okay, thank you very much, Polina. Good afternoon, dear students. I'm very glad to see you, everyone in our family, in our SCAMT family. My name is Pavel Krivoshabkin, and I'm head of CNN group. CNN is not TV channel. This is a ceramic and natural nanomaterials group. And next slide. Today, I will talk about three main directions, what we are developing in our group. First of all, this is formation of biopolymer materials with using of living organisms. Second, this is ceramic nanomaterials for medicine and biosensors. And the last theme of about systems for monitoring climate change and environmental applications. And next slide. In this case, our interest contained to knowledge about the process and mechanisms what make it possible to obtain new materials using living organisms, for example, insects, spiders, or plants, and others. And in next slide, maybe you probably know insectarium was created in our SCAMT Institute, and we can get a natural spider silk every day. And this is spider web is a unique and very interesting material with excellent excellent biocompatible properties. Next slide. And we have a lot of several methods for obtaining materials with different shapes and morphologies. For example, coatings, hydrogels, films, and ours. Next slide. Second theme about ceramic nanomaterials. We're using metal oxide nanoparticles for development of multifunctional platforms for cancer therapy and diagnostics. And we're using different types of therapy, for example, radiotherapy, magnetic therapy, and chemical therapy. And next slide. And we try of combined different treatment, for example, chemotherapy and radiotherapy, and magnetic electric therapy and radiotherapy. Next slide. These nanoparticles is a good properties for diagnostics of different types of cancers. For example, we can use optical properties, upconversion luminescence, and contrast engines for different types of tomography. And next slide, this approach we use for preparation of different types of biosensors, for example, for food monitoring, for monitoring of adaptation of life organisms, and other. And next slide. Yes, the last approach is a system for monitoring climate change and environmental applications. We use different types of devices and systems for example, for CO2 capture, storage, and reduction, for firefighting and fire prevention, for environmental application of different types of carbon hybrid materials, and our. Next slide. Thank you very much, and we are very glad to see you, chemists and biologists in our groups. Good luck.
I will thank you very much for the presentation and for the beginning of our kind of online lecture. And we are going next, and the next group is the group of Evolutionary Biomedicine. Bayezid, are you here?
Yeah. Hello, everyone. Can you hear me?
Yes, yes, we can hear you well.
Right. So I'm Bayezid Yunusbayev, and I am associate professor at Tartu University, and I will be starting a new research group here in St. Petersburg as a leading researcher. So next slide. Now, our research group is going to focus on a very interesting and important research problem of modern life sciences. It's autoimmunity. Autoimmunity is when your immune system is attacking your own tissues and cells and causes debilitating conditions. Now, if you think that it's some sort of rare disease, it's not. Later or sooner, it's gonna be related also to you as you get older, and this is why it's covered in the recent Nature portfolio. Now, the next slide. Now, why is the puzzle? The thing is that our immune system has evolved to fight with pathogens and also interact with symbionts. Now, both pathogens and symbionts, they are structurally similar. They are viruses, bacteria, and fungi. Now, the key thing here is that immune system has evolved, and we expect to see some genetic imprints in our human genome. So that's exactly that we are using to locate these genetic imprints and use this information in prioritizing genetic variants and genes that are contributing to pathogenesis. So that's briefly is called the fine mapping, and that's the first project. Next slide. So that fine mapping actually involves a lot of bioinformatic analysis. So, and it's very challenging and represents an outstanding issue of modern human genomics. Now, suppose that we have actually, prioritized and came across the gene and genetic mutation that is contributing to autoimmunity. Then the next question is to learn where these genetic variants are actually functioning, which cell type, and more importantly, what should be the physiological context that makes this genetic variant to manifest its function. Now, speaking about function, we have to remember that most of them are expected to be regulatory, so it's not easy to decipher. Now, here we also use insight from evolutionary biology. We are using pathogens to model, microbial invasion, and we expect these genes to, elicit their function. So the next slide. So that was the second project, and the last piece of puzzle of autoimmunity is the role of symbionts. Now remember what I said, that symbionts are structurally similar to pathogens that our immune system evolved to. So we wanted to understand what's going on with symbionts in our body, and the largest body or the largest population of symbionts in our body is in our intestines. So we are sequencing using metagenomics, using deep sequencing microbiome from patients, and we will be doing bioinformatic analysis to learn what's going on there, to learn about their function, their structure, and hopefully to get insight about connection between symbionts and autoimmunity and interplay with genes. Now the next slide. So this project, just 2 words about this project. Here I am hoping to collaborate with people doing 3D printing, and the idea is that to get data from white tissues. The thing is that most microbiome research currently is done based on fecal sample, and that's a problem because we only have indirect evidence about what's going on in the intestine. And the idea is to come about a small device that we can manufacture with 3D printing and use that to sample the microbiome from the right tissue. So the next slide. And as I'm only starting this research group, I will largely depend on the infrastructure of Tartu University. So if you're gonna join this group, we are going to work closely with Tartu University and use high performance computing there to do our computations and also genomic data and biobank data. So if you're interested, then reach me out. Today we will have an informal conversation around 3 p.m. I suppose. But thank you.
Thank you very much for the presentation. And we are going next to the plant genomics lab. Vladimir, are you here?
Yes, I'm here.
Okay, great. So you can start.
Okay, good afternoon, good morning, where you don't know. Actually, my name is Vladimir Brukin. I'm associate professor from Plant Genomics Group. I'm running this group, and I would like to say a few words about our groups and our research. So please, next slide, Paulina. Yeah, so this is our favorite Arabidopsis and Bokera plants. You can see on this slide. And it's one of the main object of our study. And next one, please. So actually, what study our group and general plant genetic and genomics? So first of all, of course, it's functional genomics, which study function of genes, how they function and what they're responsible for. Comparative genomics, and also we interested in sexual and asexual plant reproduction, and what is the difference and what is the gene regulates these processes. And also we study and are interested in developmental biology and evolution, how first of all reproduction or selection of gene during sexual and sexual reproduction and their fitness affect evolution of plants. So next one, Paulina, please. Our research have implementation and application, and not only our, but also plant genetics. And in our case, it's applied developmental genetics, which is a sort of basis for plant reproduction and for seed productivity in agriculture and crop productivity. And in general, so many people ask question, what for one need to study plant genomics and plant genetics? Of course, it's not only for fun, it's for understanding in general biological nature of plants, and it has a great application in agriculture, in pharmaceutics, in industry, and in production of old and new materials. Next one, please. And in our research, apart from other stuff, we use a lot of genomics and genetics approach. We kind of focused on genome assembly, gene annotation. It means to know architecture of genome. And we use data produced by whole genome sequences. It's Illumina, short reads, PacBio, Nanopore, HiC, and other sequencing platforms. So, and we use it for, as I told you, for genome assembly, for genome annotation, identifying different genome features, genome comparison for comparative genetics, and we discover new gene and we use it for functional genetic. Also, we apply fluorescence sensitive hybridization of the genes to physically locate loci and genes on the interesting chromosome. And of course, we do phenotyping of our lines which we study. Next one, please. Okay, so within plant genome assembly project, we use, as I told you, comparative analysis of plant genomes depending on the way of reproduction. And in these slides, you see difference. It's so-called chimer analysis, where we show on the left it's a low heterozygosity plant and a high heterozygosity plant. So, for example, plants which reproduce asexually, they often arise as old and very complicated hybrids. And most of their genomes are highly heterozygous, which you see here in the right slide, in the right insert. So next one, please. We also do comparative analysis in plants. As I told you, many asexual plants, they're very long and very sophisticated hybrids. And here, for example, you see slides pairwise comparative genome in two species, which parental lines for some apomictic line in Berkera. And we try to find what is the percentage of each plant who is an ancestor plant in this hybrid. So next one, please. Yeah, and for our research and for phylogenetic analysis, we also use a protoplast, and here you see chloroplast, one of the type of protoplast, for genome assembly of chloroplast and genome annotation for using it for further phylogenetic analysis. And here it's indicated small fragment, highly variable, but at the same time very conserved, called TRNLF region, and it's used for phylogenetic reconstruction. And in fact, chloroplasts, they inherited only by maternal lines via cytoplasm, and they use as a sort of haplogroup of plant. And next one, please. Yeah. For identification of chromosome blocks in ancestral progeny of in ancestral lines of certain plants. Here, for example, in this slide, it showed Brassicacia and several ancestral blocks. And we try to relocation of these blocks, including synteny blocks and their location in our genome study. And the study, we use fish fluorescence in situ hybridization and GISH, genome in situ hybridization, to see where these particular genes are located. So here you see two genome of ancestral lines in their progeny in hybrid line, different colors, yellow and green. And next one, please. So also we study, as I told you, asexual reproduction, and one of the main representative of asexuality is apomixis, which is a production of plants or offsprings via sexually produced seeds. So they bypassed meiosis, and in fact their progeny or offspring, they're genetically identical to parental plants. And it's very important for evolution on fixing genetic traits in agriculture. And unfortunately, molecular mechanism is not very well understood so far. And we try to understand components that regulate apomixis, and also their role in breeding and evolution. And also we use it for molecular and evolutionary study, and mechanisms that control apomixis by comparative genomic study. So we try to find genes and gene loci which involved in switching these persons up and back. So next one, please. Yeah, and one of the application of apomixis, in case it will be introduced in the culture plants, could be what you see here. It's a heterosis. When you cross pure lines, you see so-called flash of hybrid vigor, which unfortunately only in first generation of normal sexual plants, and usually segregate away in the next generations. But in case of apomixis, it could be indefinite generation, which combines this useful trait. And next one, please. So this is slide show kind of agricultural advantages of apomixis. On the left, you see sexual reproduction when due to meiosis and meiotic recombination, all useful traits which selectioner produced for many years, they segregate away after crossing, after first generation. But in hybrids, as I told you, see no segregation, no meiosis, and therefore all traits are retained in the next generation. Next one, please. So, also very important project which we run, it's phylogenetic and functional analysis of gene which associated with reproduction in the representative of Bockera genus, very interesting genus from Brassicaceae family, and it's close relative to Apomixis, for which functional annotation and genome database are available. And we study also genomic and transcriptomic differences of apomictic and sexual lines. We perform functional analysis and phylogenetic analysis. We look for additional alleles, gene which are candidate for associated with apomixis. And we study characterized genome of sexual and asexual lines, genome structure. We're looking for features like copy number variation, single nucleotide polymorphism, indel means insertion and deletion. And we're also looking for gene ontology classes for special gene under interest and their enrichment with apomictic alleles. We do a comparative expression of genes and also cloning-based candidates to analyze them by complementation in Arabidopsis. And next one. Yeah, as I told you, we use phylogenetic analysis. Here it's phylogenetic tree for gene locus is Apolla, one of the main gene in Bockera which associated with apomixis. It has a kind of biallelic inheritance type. And he has upper and sex alleles, and combination of this allele produce plants or hybrids with different type of reproduction. So, and we study evolution of this and other genes by phylogenetic analysis. And next one. Okay, and here I'm in fact actually done. And here it's our collaborators. We collaborate with different countries, with a lab from different countries, from Zurich University, where I used to work for many years, Czech Republic, University in Brno, and also in Turkey, in Indian Institute of Technology in Madras, and we use with States in Florida Museum and University of Florida, and also a number of institutions here, just to say a few. It's St. Petersburg State University, Botanical Institute of Russian Academy of Science, and Kurchatov Institute. And that's it. And if you're interested more, if you're physically here, you can come and visit us today from 3 to 6 o'clock. And I wish you good start in SCAMT. Thanks.
Thank you, Vladimir, very much for your presentation. And we are moving to the next group. That is, just a second, that is a Neuroeducation group. Mikhail, are you here?
Yes, I'm here.
Okay, glad.
And happy to see so many new master students. I am Dr. Mikhail Korushkin. I represent the Cognitive and Educational Neuroscience Lab. It originates in chemistry and biology cluster, because I came here as a chemistry educator, and like in a broad sense, the lab is dedicated to research-based approaches towards making a positive change in chemistry education, or maybe life sciences education. And we have two main research areas, which are investigating the effectiveness of various educational methods, including but not limited to virtual reality, augmented reality, different online modules. And the second research area that we represent is investigating the role of individual differences, such as intelligence, spatial ability, creativity, and things like that. Next slide, please. So let me dive deep into the first research area that we offer. Basically, it is dedicated to the following thing. When we learn or teach or have an education experience, we have different learning environments. And it's always a very good question how you actually create the best learning environment possible for a group of learners. And in order to do that, we employ various experimental methods, like, for example, eye tracking technology, because the way your eyes behave while you're learning actually tell educators a lot about how to better create the proper learning environment. For example, we have a Russian Science Foundation research grant where we use eye tracking in virtual reality and chemistry education in order to evaporate the borderline between actually the learning and the evaluation process, because usually this evaluation takes some separate time, and we want to combine those processes. And there are a lot of various different projects in this area, the ongoing ones that correspond to more or less to virtual reality and immersive molecular dynamics, eye tracking, electroencephalography. And let us move on to the second research area. The second research area basically answers the following question. Every learner has his or her own individuality, of course. And the question is, how do you tailor this education to the needs of this individual being? That is the important question. Because the first question is still more of a kind of a group thing, learning environment for a group of learners, whereas this second research area is more dedicated to individualized learning for an individual. And in that area, we have a very new postdoc who came from Sirius University of Science and Technology with his project investigating the role of spatial ability in chemistry education. Because, you know, spatial ability is the ability of how you can get accustomed to different spatial, you know, correspondence of different objects. And it's very important for a chemist because, you know, there are different isomers of various kinds, and a chemist needs to know how to operate with the different objects of different chirality, different complexity. So in those areas, we employ electroencephalography, for example, to measure experimentally the cognitive abilities of future chemists. And these are the two research areas. But I want to stress out that, of course, new projects may emerge and be triggered by your coming to our research group. And everyone basically does his or her own project, like it's 90% of your own work. And going to the final slide, I'd like to say that we organize and cultivate our research the following the following way. Either myself or our postdoc, Dr. Maxim Likhanov, we oversee the projects. Also, we have research professor, our scientific advisor, Yulia Kovas, who also is going to supervise herself some of the projects, and she's going to become a research professor quite soon at ITMO University. So in case you have a passion towards making a positive change in chemistry education, I'm really happy to welcome you. The only thing is that apart from that passion is to just be really ready to take your responsibility and do quite a lot of hard work to, you know, learn something new. Maybe go to some of our universities where we have collaborations, like Tomsk State University, Sirius University of Science and Technology, Higher School of Economics, because we have a big network of different experts. So we'll be really happy to see you today between 3 to 6 p.m. or any other day. I'm very approachable. You can always, you know, give me a call. And thanks for your attention.
Thank you very much. And yeah, we continue now. And we continue to one unusual presentation, as one of our group leaders of the group of sustainable electrochemistry is actually in Canada, and you know it's hard sometimes because of time difference to organize all these meetings. So she recorded the video for you, and I hope that you will be able to hear the sound. So let's try, and if something's wrong and you will not hear the sound, please tell me.
Hi, my name is Anna Klinkova, and I'm a team leader of the Sustainable Electrochemistry Group. Our research is focused on reactions and catalysts for electrochemical conversion of carbon dioxide, wastewater contaminants, and biomass to useful chemicals. In general, the development of renewable electricity-powered electrosynthetic processes for the production of high-valued chemicals can contribute to the reduction of chemical industry's carbon footprint. In our approach, we specifically explore the utilization of waste as precursors for electrosynthesis to further increase sustainability and benefits to the environment. A couple of examples of such transformations include electrochemical coupling of carbon dioxide with an organic precursor to produce ibuprofen, or wastewater ammonia and urea electrodecomposition to generate hydrogen fuel, or conversion of biomass pyrolysis byproducts to a nylon precursor. To facilitate these transformations, we studied the performance of well-defined nanoscale electrocatalysts targeting high activity and selectivity in the reactions of interest. We synthesized these well-defined nanostructures using a variety of experimental techniques. We then evaluate their performance in electrochemical reactions and study reaction mechanisms by analyzing current efficiency, stability, and product yields. This work is done together with my team at the University of Waterloo in Canada, where I am primarily located. The team at ITMO is co-supervised with Dr. Kriva Shapkina, who advises students on site. We conduct joint monthly conferences between the ITMO and the Waterloo teams, and all the members closely collaborate and help each other on the weekly basis. Please reach out to me if you have any questions. Thank you and have a great day.
And I hope that you could hear Anna well. And we are moving to the next group, that is Biofunctional Materials group that will be presented by Artur Prilevsky. Artur, are you here?
Yep.
Okay, great. So I give a word to you.
Okay, good day, dear new students. A very brief talk of our brand new group called Biofunctional Materials Group here in SCAMT. So the first one, who we are. Next, please. Polina? Yeah, next. Okay, so lab head is Professor Pavel Lövkin from Karlsruhe Institute of Technology, Germany. Pavel is a world-leading specialist in different scanning technologies in organic chemistry, and he have a lot of published papers in good journals like Advanced Materials, Nature Communications, and so on. He also co-founder of two companies in Germany, which specialized in different scanning applications. Next one. Here's me. I'm a postdoc in SCAMT Institute. I have a PhD in biology and specialist in different applications with nanoparticles, nanotoxicology, and so on. Have some good papers in different areas connected mainly with biomedical applications. But the most important question is what we are in our background. Next. So probably every one of you sees some super hydrophobic or just hydrophobic surfaces. It's basically surfaces that can reflect different liquids. And these examples of the surfaces, substance which were developed by Pavel in Germany and can be applied to almost any surfaces. But what if we will go next? and add some structuring to the surfaces. For example, here is a super hydrophobic coating of the glass and some spots left without any coatings. And if we'll go further, we will be able to physically divide some droplets on the surface. And with this approach, we can, and with this approach, we can create what is called droplet macro array. So this droplet macro array incorporates thousands of physically divided dots, and in each of these dots can be done something. So next one. But what are we going to do here in SCAMT? Next. We're going to move from hydrophobic coatings to omniphobic coatings. Omniphobic coating is something that incorporates both features of oleophobic and hydrophobic. So they have some common features with hydrophobic coatings like scalability, easy to obtain, can be applied to any surface and so on, but it's also have some advanced features like versatility, reusability, and so on. Next one. So what this droplet microarrays and this technology can be applied to? So it's both fundamental application and well, some applied science. So in fundamental science, you can use these droplet microarrays to study, for example, interaction of cell spheroids, formation of cell spheroids, mechanism of cell proliferation, cell metastasis, cancer metastasis, cellular differentiation, and so on. You can study formation of biofilm bridges, bacteria migrations, gene transfer, and different other applications that can be considered as fundamental. Next one. If we will go further to some applied approaches, it's different laboratory screening techniques and point-of-care applications. So you can use, for example, a biopsy from a patient. So take some cancer cells from a human and test some drugs, anti-cancer drugs, for example, to understand which drug is best used to treat this patient. So basically, it's unlimited number of different approaches that you can imagine using this droplet microarray technique. Next one. But who are we looking for? So what you should have. As I mentioned, we are a brand new lab, so it's our first year here in SCAMT. And that's why you should be ready for a lot of independent work, a lot of analysis of scientific literature, planning of experiments, and most important, ready for organization work in a new laboratory, arrangement of lab routine, and of course, ready for internships and so on. And about the knowledge which is desirable in our lab, it's organic chemistry, it's biology, and if you have some programming background, it would be very nice. But it's our demands, and what are we offering in exchange? So it's well, both improvement in hard skills and soft skills. So of course it's will be English improved because you will work with foreign researchers. You will have some definitely will have some internships in Germany. And of course, it's some organic physical chemistry, cell culture experience, and so on. For many students, as I know, it's very important to have some skills, some get familiar to work with industry and clinic, because our platform can be easily, let's say, introduced in different lab routine, for example, such companies as Hemotest or in vitro or different clinical application. So we hope to go to this area and get familiar with this one. So you can contact me today or any day. So I will be glad to answer any additional questions.
Thank you very much for the presentation. And we are moving to the next lab. That is Inkjet Printing Lab. And Alexander, are you here?
Yes, yes.
Great.
Hello, everyone. Hello, everyone, and I'm glad to introduce Inkjet Printing Lab. And I'm really glad that today I'm ready to represent your very interesting and highly technical approach to get nanoarchitectures and microstructures. The main concept of concept of Inkjet of Inkjet group is to develop the very cheap and very accessible approach to fabricate nano architectures into the different surfaces and from the different substances. So the main idea is the following. Everybody knows that nanolithography, like ion beam or laser beam, it's quite common to get a nano architecture. And everybody knows that this approach are extremely expensive and non-accessible to high surface applications, for example. And the idea is the following: if we had a very simple and very cheap approach like inkjet printing device in a desktop, for example, we could achieve absolutely the same nano architecture. But the key question, how can we do it? And my group, who works in the focus of inkjet, possess the some programming and some engineering approach to modeling the process from single drop to nano architecture, and we design different properties and different self-organization processing from one drop to the surfaces. Now, Polina, please, next slide. And now you can see how can we do it. Here comes some examples. We have a system that one student can realize individual project by himself. And here comes, you can see that we made some publication during for 1 year and published it into the top journals like Advanced Functional Materials or other top journals. And Polina, please, next slide. And here comes, you can see that now we have a free independent research grants supported by Russian science foundations. 2 of these grants are supported individual groups, and 1 grant supported the small group for, with head of young postdoc. And the focus of these grants are the following. The first one is the memristor fabrication by inkjet technique. Memristors is a small devices for intercellular detection, for intercellular operation, and strong the hypothesis and algorithms for cells communication. The second project is approaches for the fabrication and printing nonlinear optical nanostructure. And the last project, one of the biggest project from my lab, are in the focus of inkjet printing of optical nanostructures and light emitting nanostructures. Now we had a slot for four students. On the right side, you can see the first one is the inkjet printing for nonlinear optical nanocrystals. The second one is a very, very ambitious project for the inkjet printing of light emitting diodes from the gallium phosphide and arsenicum nitride. And the third one is inkjet printing of optical holograms, the first example of inkjet printed holograms, and the fourth one is printing of perovskite with the high light emitting effects. Thank you and welcome to Inkjet Group.
Thank you very much for the presentation. And we are moving to the next group, that is Molecular Robotics and Biosensor Materials. LD, are you here?
Hello, yes.
Okay, great. So you are here and you can start the presentation.
Yes, I'm here, but the problem is that there is problem with audio here. Okay. Hello, can I start now?
Yes, yes,
Okay.
you can
Hello,
start.
everybody. My name is Eldib. I'm from the Lab of Molecular Robotics and Biosensors. We can go to the second slide, please. We work in our lab in two directions, two main directions: therapeutic direction and diagnostic direction. The therapeutic direction, mainly for treating cancer, we are using a lot of interesting approaches like DNA zyme in gene silencing, antisense system in gene silencing and small interference RNA. Next slide, please. While the second direction, we work with developing DNA machines, really so sophisticated and interesting DNA machines, also based on the DNA zyme technology. And you will know more about all of these technologies when you will visit us to our lab today, I think. When you will visit us to our lab, and we will talk more in details about that. We developed DNA machines for detection of viral RNA without any kind of amplification, which is something new till now. And also we developed another DNA machines for detection of the double-stranded bacterial DNA and viral DNA. Next slide, please. Our lab consists of 22 group members, including 6 PhD students who will always be in your help in any time you will be you will need any of them, or you will need to ask any question. Also, we have another 15 well-trained second-year master students who also are going to be supporting you anytime you are in the lab, and they are going to be showing you anything you need in the lab. Next slide, please. Our lab have our main leaders, Professor Dmitry Kolpashikov, who is so supportive professor. Always you can reach him by Skype. He always welcome any new idea, always going to be helping you. While we have the main leader of the diagnostics group, Mrs. Maria Rubel, which is also really so good a woman that you will meet in our lab. She will help you, whoever team you are. She's always caring about everybody, and she has from the knowledge which really can feel a lot of, which really can be enough so much for you. She's working with Professor Kolpashikov more than 3 years, while the main leader of the therapeutic group, Mrs. Daria Nadrezova, she's working with Professor Kolpashikov for 5 years. She is the main leader of the therapeutic group, and she is responsible for all our experiments which we conduct in the cell lab. Next slide, please. We work in connection we do our work in connections with other laboratories and other foundations, like Institute Pasteur, like Institute of Cytology, like Institute of Influenza, Institute of Children Infection, and St. Petersburg State University. We have 2 grants, RFI and CSP, and thank you so much for your attention.
Thank you very much for the presentation. And now we are moving to the industrial lab. And we again meet Alexander.
Again, thank you very much for your introduction. Now I'm still in my office own, and I will be in more focus when I get the presentation. So industrial loop, what is the key idea? So we had 14 labs in our department, and every lab are in the focus, of course, more research than applied project. And the answer to make their such type of group, industrial group, is to support the intern labs activity and support the interdisciplinary projects, from theoretician, for example, to printing, and then to sell the technology, for example. And what can you receive? Polina, please, next slide. What can you receive then you join to industrial group? What we are doing inside of our groups, and what we can do it future. For example, we are creating the startups. Now we have the 4 open startup for different projects, and now we have the 18 individual project which are successfully commercialized. We are participate and win business grants and first round of investment, for example. We also made the legalization and packaging of the product which we prepared by ourselves. And we also work in the different markets, for example, B2B, B2C, and B2G. Polina, please, next slide. The group of industrial lab is a soft skills and hard skills students. They are represented on this slide. They made a lot from science and from proof of concept to direct contact with the customers and consumers. Polina, please, next slide. So what we are doing right now, we started 2 years ago, and we developed the first we already implement this technology to the market. I mean the fabrication, fast hardening form for firefighting and fire prevention. It was the first example in the world for making hardening foam with the first class of biodegradability, and we also prepared the product with a very high economical effects. So this technology already implemented into the market. Now, then we moved to the vehicle industry, and we had 8 independent product for vehicle industry. We also made 2 projects in the focus of therapy and prophylactic effects for skincare, for example, effects. We also fabricated B2B product. I mean, like titanium aerogels for optical applications, and we exported every month 20 kilos. Yes, the 20 kilos of this product to China and America. And now we had our financial supportance of high-tech and deep-tech technology, and now we are in the focus of high-tech products and high-tech projects, which are relevant for unicorn the technology, for example. Polina, please, next slide. And when you join to the industrial group, you can follow to creating your personal startup, for example. You can attract and see the investments from the investors. You can cover the patent technology, the IPO technology, and work with the real markets. I mean the marketplaces, I mean the consumers by yourself. And the working with the industrial group is not quite simple, because you need to cover your scientific activity, you need to cover your educational program, you need to cover and interact with the big bosses and with their very aggressive markets, for example. So, but at the end, you will be as an engineering, as a good manager, and you will possess a very high and good soft skills. Okay, and Polina, next slide. Thank you for your attention. And if you are gonna build a unicorn by yourself, please come to industrial group. We can make it together.
Thank you very much for the presentation of industrial group. And we are moving to the next group. Marina, are you here?
Yes, I'm here. Hi, Marina. Thank you.
Okay.
So my name is Marina Dukhinova. I'm a PhD and the leader of cell biophysics groups. I think that you all know that biomedicine is not only about the cells, tissues, and organisms, but also about how physical and chemical factors can impact our cells and our body in general. Next slide, please. Okay, so here in our group, we are interested in two major directions, which are oncology and immunology. We study how different external and internal factors can influence cell behavior and cell molecular signaling and their transcriptional activities. And the next slide, please. Yes, so among the internal factor, we are mostly address the aspects of radiotherapy, chemotherapy, and cell microenvironment in terms of both oncological and immunological research. And then for cell destiny and functionality, we look at the mechanism of how transcriptional regulation can impact cell behavior. Next slide, please. Yes, we have a broad direction of immunological projects which are relevant to fundamental immunology and transcriptional regulation of different cells, such as mononuclear macrophages and lymphocytes, and with particular emphasis in antiviral immunity and anti-cancer immunity. Next slide, please. We have a number of publications which you can address if you're interested in our research. The next slide. This is our team and the projects. And by the way, most of the research are now run by young scientists, PhD and MPhil students. And we are expecting you to be among them by next year already. And all these projects are done in a broad scientific collaboration network, as well as in Russia and worldwide. And welcome to SCAMT, and thank you for your attention.
Marina, thank you very much. And we are moving to the next group, that is Nanopharmacy group. And Alexandra, are you here?
I am here. Do you hear me?
Great. You can start.
Okay,
yes.
So I ask you to move slides. Yeah, am I right, Polina? Thank you. So hello, my name is Alexandra. I'm a PhD student of Nanopharmacy group, and I will be happy to introduce you our group. And so the main idea of our group, the main postulate that we are not doing like regular research. We are moving to frontiers. We are looking for frontiers. We are looking for breakthrough research to break up scientific society with the best concepts. So the next slide. And I believe the first branch of our lab, and it is my branch, it's liquid metals. They are special materials with special properties that are liquid at room temperature, and they possess a lot of unique properties like high electric conductivity paired with fluidity, flexibility. They are non-toxic compared to mercury, and they obviously possess low melting points. So last year we developed a unique methodology. Polina, please, next slide. Thank you. We developed unique methodology of producing liquid metal nanoparticles, and from these liquid metal nanoparticles we can produce hollow multimetallic capsules that can apply it in every field of science. So this is the basic technology we developed. So the basic technology leads to a lot of new projects, and the first one is anode materials for batteries. This project is already started, so we are welcoming students to join this project who are interested in lithium-ion batteries of new storage, new energy storage materials, and so on. The next project is about, can you, thank you. Yes, thanks. The next project is about shape controlling, because this is a new trend in nanotechnology and nanopharmacy too. The shape controlling properties, the shape controlling procedures are highly requirable for every nanomaterial. So the following project is about the physical and chemical methods of shape controlling, and we are going to produce the first case. We are going to make the first case of liquid metal aerogels. So it's going to be the first case of production of these aerogels, and we are welcoming students for this project. Well, stuff about me. Let's talk about me. So the next slide, Pavina. Another branch, big branch of science in our lab is artificial intelligence. There are people who are applying machine learning and strategies and all this artificial intelligence strategies to predict shape and size of nanomaterials, to model nanomaterials in one computer and so on, to model the nanozymes properties, to predict the properties, chemical and physical properties of nanomaterials. This is a new trend. This is a really frontier in modern scientific society, and there are two people, Nikita and Yulia, that nowadays moving science in this branch. So we are welcoming students who are interested in artificial intelligence and machine learning to join Nikita and Yulia and to make their own contribution in this frontier. The next slide, please. And the last big branch of our lab is magnetically controlled biochemical processes and systems. This is about a lot of materials. This is about magnetite nanoparticles. This is about other magnetic nanoparticles. This is about magnetic biorobots, magnetic materials that can be applied as non-invasive surgical tools for a lot of applications like thrombolysis and so on. It's about new fundamental problems of magnetic levitation. Can we use artificial magnetic levitation and for controlling properties of magnetic materials, magnetic nanomaterials, or bulk materials, and so there are several projects. They're about magnetic levitation systems, where guys grow big crystal moths in magnetic levitation. So these moths are like flying in the air, but it's all about science of magnetic levitation. They're about biotechnology too. It's about to make materials that can be applied in biotechnology via controlling their magnetical properties. They're about a lot of cool systems that can be controlled by external magnetic fields. So there's members of this mini group in our Nanopharmacy group, Daniel, Anastasia, and Anna. They already developed their own projects and specialists in this field. So we are welcoming students to join them who are interested in magnetically controlled materials. And I believe that's all. Thank you for your attention. Join the Nanopharmacy group.
Thank you very much, Alexandra. And we are moving to the next group. That is TheoMAT group. Ivan, are you here?
Yep. Can you hear me?
Okay. Yes, I can hear and see you. You can start.
Okay. Hi, everyone. My name is Ivan Chernyshov, and I'm a postdoc of theoretical chemistry group led by Professor Pitko. But before talking about our specializations, let me explain briefly what theoretical chemistry is. The first goal of this science is to describe chemical systems and processes. In other words, it's answering the questions like, why does this substance possess such properties? Or why does this reaction go this way? And the answers to these questions are theoretical models, which are required to achieve the second goal of the theoretical chemistry, to make predictions, like how to modify this material to enhance its properties, or what are the optimal initial concentration for this synthesis. Theoretical chemists use physical and mathematical modeling tools like quantum mechanics or quantum chemistry, molecular dynamics, machine learning, and so on. And therefore, our main instrument is a computer, so we can easily work from home. Now let's return to our specializations. Polina, next slide, please. Our main scientific topic is the modeling of real solutions. Why is that so interesting and important? Quantum chemistry is very good in predictions of gas phase chemistry, where molecules are isolated. But when it comes to solutions, the model molecules are surrounded by solvent molecules, and this solvent shell is always in dynamics. And there are some empirical methods in theoretical chemistry that can model ideal or highly diluted solutions, but they actually cannot consider the effect of other regions and therefore to describe concentrated solutions. The possible way to describe them is to explicitly consider all the solvent molecules and to use the state-of-the-art methods like QMMM or ab initio molecular dynamics. But these methods require a huge amount of computation time. So you can use them to model usually only one system. And you cannot use them to solve problems like optimization of the initial concentration of regions in some synthesis, yes. And that's why we developed new methodologies to describe and predict the effect of media on various chemical processes. And we apply them to various industrial catalytic processes and synthesis of new materials. Next slide. In addition to chemical modeling, we use programming to transform our developed methodologies into the programs to facilitate automation in future research. Next slide. Also, we use machine learning and, strictly speaking, methods of cheminformatics to create so-called knowledge-based predictive models, or to parameterize various empiric physical models. In addition to the indicated areas, in our group, you can do anything related to theoretical chemistry, and the only limitation here is the competencies of your supervisors. It is difficult to reveal more details of our current and future research in 5 minutes only. So if you are interested in working with us, if you like theoretical chemistry, come to our group today to chat. That's all. Thank you.
Thank you very much. We are super close to the end of our meeting. There are 3 labs left, and we are moving to the next one. That is 3D printing of functional biomaterials. And Yulia, are you here?
Yes, I'm here. Do you
Great.
hear? Yeah. Yes. Okay. Hello, dear students. I'm very glad to talk about our lab. My name is Yulia Pilipenko, and I'm a postdoc researcher in the group of 3D printing of functional materials. So our lab is headed by Dr. Yelena Kryashapkina. And since 2019, we have the project, which is supported by Mega Grant Foundation, and the head of this grant is Professor Eugenia Kumachova. She's a very famous in polymer world scientist. She is located at the University of Toronto. And we have a close connection to advance our facilities and to adopt new technologies from Eugenia's group. So, okay, Next slide, please. Yes. So in our lab, we always start from the materials for 3D printing process. And among the materials we use biopolymers, namely cellulose nanocrystals and chitosan. And we synthesize different shapes of synthetic polymers, like using controllable radical polymerization. Using this technique, we can obtain different kind of architectures, for example, block and graphical polymers with smart properties. So next slide. Yes, and we have, as the production method, we use 3D printing to obtain the materials with different flexibility, size, rigidity, and even multi-layer structures for biomedical or cosmetic purposes, or even smart packaging and security printing. Next slide. And here are several examples of our work. So here we design hydrogel based on cellulose nanocrystals and gelatin, which were decorated by carbon dots. And this hydrogel was optically active, and it has a slight antibacterial activity against 3 different strains of bacteria. And using this hydrogel, we can obtain optical response during the treatment process. Next slide. And in this project, we develop self-healing wound dressings with sensorial property. Using 3D printing, let us to obtain different materials with specified parameters without affecting their unique properties. And here we design chitosan cross-linked hydrogels with incorporated pH sensor dye, and we printed those hydrogels using to obtain this kind of holes to encapsulate the healing agent like growth factor for better wound healing. Next slide. Yes, and we can use 3D printing process to protect products from counterfeiting, for example. And we here we obtain hydrogels with fluorescence in different ranges of visible light. And as you can see here, with this under UV irradiation and visible light, this hydrogel are totally transparent at daylight, but it fluorescent at UV light. Next slide. And so this hydrogel can be printed in different forms and shapes, and with different colors, and even multi-layered structures like those fishes. And this property can be used in a smart packaging in food industry, for example, with indication of expiration date of products. So next slide. This is all about our projects, and I want to say that we have a really friendly and comfortable atmosphere in our group. We have several master students, second year, and PhD students for your to help you. So if you are interested in polymer science and 3D printing process, welcome. Thank you for attention. That's it.
Thank you very much, Julia, for the presentation. And we are going next to the microbiology group. And Sonia, are you here?
Yes, I'm here.
Okay, great.
Okay, so our group leader is Yelena Kosher. She got her PhD in microbiology and worked with Yersinia pestis. So she is a great specialist in microbiology. And my name is Sophia, and I'm her PhD student. And I'm working about 3 years in SCAMT. So we can go to the next slide. In our laboratory, we have a big and very friendly group, and the projects of each participant of a group is devoted to the to the study of different ways to manage microbial communities and to the rule microbial life, to use microbes for applications in the fields of agrobiology and medicine. So we go next, and and here is my own project that I managed in my master's degree. We found a metabolic stimulator from gut microbiota. This is a polysaccharide from Enterobacteriaceae. And this polysaccharide have different interesting properties. They're about energetic metabolism. And we also make a big research in microbiota now. We work with medical organizations, and we also published, for example, this review, and we have another one review that we already submitted. So speaking about metabolic stimulator, we can go to the next slide. We studied its properties in different cell lines, in cancer cell lines and normal cell lines. We found that metabolic stimulator affects organization of mitochondrial network inside the cell. So mitochondria became more condensated inside the cell, and also some cells like murine cardiomyocytes cell line, they became more differentiated. So this is basically very interesting, because usually we think that polysaccharide can be just digested, but we found that they have a huge regulator function. So if you go next slide, you can turn on the video on the slide. So we also tested this stimulator on mice, and here is the most interesting thing. We found that this stimulator strongly increased mice activity. So as you see here, the normal mice is in the house, and the stimulated mice, she's jumping, climbing, all of this stuff. We also tested mice behavior in water and found that mice had increased efficiency in aerobic exercises. So it's very interesting to continue studying of these polysaccharides, and we are going to try to manage microbial community to facilitate production of this polysaccharide. So we go next. Another way to touch the activity of microbial communities is to use different biotic and abiotic materials to regulate it. So, for example, we construct with a group of Nanopharmacy different nanocomposites that are active against biofilms. So, nanocomposite basically is something like, for example, container that can take some, for example, drugs inside the nanoparticles with the use of magnets. Also, we can use some, for example, gels with modified properties with, for example, some antibacterial agent, and inside the gel it becomes more active, and it can be very effective against, for example, chronic wounds. Also, we can use not only such inorganic and classical nanomaterials, but we also can use biotic molecules. So, for example, DNA enzymes, about which you have heard recently. So we use DNA enzymes to test it against resistant Escherichia coli, also against Staphylococcus and against Coxsackie virus. Next. So yes, another example is when we combine abiotic nanomaterials and biotic organisms. So in this project, we used gel nanomaterial and inserted lactobacilli inside of it. So lactobacilli are natural producers of bacteriocins. It's a molecule of bacteria that is active against other bacteria. So this material became very, very active against Staphylococcus and Escherichia. So it's very promising to use it again in living system against different chronic wounds and maybe some burns. And next slide. So we tested this hybrid living material against against burn wound in mice. So we had the gel of Nipam and Lactobacillus again. And next slide. As you see on the picture, we have seen that this material increased and facilitated the rates of healing of this wound. So again, we made a very active material that consists of inorganic material and living lactobacillus that facilitated healing of a wound. This is a new approach in biology and modern science in development for example, healing bondage. Next slide. So those we were talking about materials, and we also can use bacteria as our material. So we can use genetic engineering manipulations to change properties of bacteria. So for example, we used probiotic Escherichia coli and inserted genes inside of it to make it possible for bacteria to insert inside the eukaryotic cell and deliver some therapeutic materials. For example, it can be used to deliver some proteins, some silencing RNAs, and we are developing this strain basically just to increase production of apoptotic protein, to increase apoptosis in the cancer cell line, and this will be very specific only for cancer cell lines and will not affect normal line. So this is a new approach in delivery of shuttle vectors into cancer cell lines. So next. And the last, we are trying to manage not only single bacterium and single nanomaterials, for example, but we are also trying to design microbial consortia to manage properties of the whole systems. For example, we can develop an artificial microbial consortia to manage properties of soil and soil-plant interaction. And maybe this will become a new direction in our projects if some occasions will happen. So I think that's all. Now, the last slide. Yes, so welcome to SCAMT and our laboratory. We will manage and rule microbes and microbial communities.
Thank you very much, Sonia, for the presentation. And we are moving to the last presentation for today from the labs, the presentation of Applied Genomics Lab. Polina, are you here?
Yeah, I'm here. Hello.
Okay, you can start.
Okay. Good afternoon, dear future colleagues. Today I would like to introduce you the Applied Genomics Laboratory under the guidance of Alexey Komissarov. Next slide, please. The current slide demonstrates our team. It consists of the second-year master students and from the 4 PhD students. Next. Our laboratory starts 2 years ago from an empty room, and we create the atmosphere of our laboratory and all the stuffs in it by ourselves. And the next slide demonstrates how it looks like for now. Next, please. We try to combine different types of education and working on project, and among them are work in our laboratory and hackathons in which we solve the huge task by all our teams. And to build our atmosphere during this. In this year, we got an experience of graduating the master students, and the 4th of them are now the PhD students in the Applied Genomics Laboratory. The projects are related to 3 main tracks. Among them are software building, genomics bioinformatics, and structural bioinformatics. Each of these directions has its own curators, and you can choose between them based on your experience and your skills. For instance, if you want to develop some software or create your own databases and you have an idea how to do it, we can help you in them, and we also can help you to learn how to do it and to obtain the necessary skills if you haven't them, but if you have the motivation. The next group of projects is genomic bioinformatics and comparative genomics. This is the best direction if you are a biologist and would like to study some interaction between organisms or to study its evolution. And if you are like to write articles and you are absolutely in love with science. We have some plans for a future to create a genome assemblies of rare species. Among them are some lizards, insects, and animals. During the future next month, we will start them. And the atmosphere with our lab, as you understand, is very comfortable and soft. But I would like to say some words about the newest direction of our laboratory, is structural bioinformatics and machine learning. If you want to obtain skills how to create your own networks, how to work with machine learning and applied artificial intelligence, you can come here and ask these people for more information. Yeah, it may help you to, for instance, discover new medicine or new drugs for cancer treatment and for treatment of other diseases. All of our projects are now divided on fundamental part. Here you can see the current project which are existing now, applied project, and some tools which was created during the last 2 years. You can see them on our GitHub. And you also can suggest your own project, and we integrate you into one of these 3 directions. We're absolutely in love with our laboratory, and you know more about it from the YouTube video, but it's also in Russian. It's long speaking of our supervisor in Sirius, Sochi. We are waiting for you in our laboratory, waiting for you offline, and enjoy the silence.